Can you hear me? Oh, I can hear it now. All right. Good evening, everyone. Seriously, every year we have to do this? Good evening, everyone. I mean, that's not that hard, is it? Well, thank you all for joining us today. There are all kinds of seats all over the place. So come on in and have a seat. This is the 8th year for Postdoc Slam. That by itself is just worth a round of applause. And we still have people, we still have postdocs wanting to participate in Postdoc Slam, and we still have you here to support them. For those of you who don't know, I'm Nikkei Blake. I have the honor of serving as Dean of the Division of Graduate Education and Postdoctoral Affairs, and GEPA for short. That's what we affectionately call ourselves, GEPA. Not GIPA, GEPA. So we have an audience here, and we have some folks in Zoom, our live stream, watching this as well. So I just want to welcome you all. I am not going to be before you very long because I know you didn't come to listen to me. Let's just start with our land acknowledgement. As we gather here in person on our campus in San Francisco, we want to acknowledge the Ramaytush Ohlone people who are the traditional custodians of this land. We pay our respects to the Ramaytush Ohlone elders past, present, and future who call this place, the land that UCSF sits upon, their home. We are proud to continue their tradition of coming together and growing as a community. We thank the Ramaytush Ohlone community for their stewardship and support, and we look forward to strengthening our ties as we continue our relationship of mutual respect and understanding. All right. I want to just take a moment, as we do every year, to recognize and celebrate the incredible contributions of postdocs who make our community such a wonderful, vibrant place. So, as we do, we're going to embarrass you, and you can't sit if you're not a postdoc. So if you're a postdoc, please stand. Everyone, if you're a postdoc in the room, please stand. All right. Now keep standing. Now you hear the love? Are you feeling the love? Yes? All right. Now, if you are sitting next to or in the vicinity of a postdoc, no, don't sit, keep standing. Just turn around, give them a high five, and say thank you. Awesome. See people walking up and down the aisle. I love it. All right. Now, Postdoc Slam, for those of you, if this is your first time, Postdoc Slam is part of UCSF's celebration of postdocs in the National Postdoc Association Week. How many of you have been to events already this week? No one. No one was out there for lunch this week? No one? No. Thank you. That was a postdoc appreciation. Okay, clearly we need to communicate better. That's why we're going to learn from these guys today. So Postdoc Slam is the featured event for a National Postdoc Association Week, Appreciation Week celebration. And seriously, on a very, very serious note, postdocs are an invaluable part of the UCSF community. I like to say that they are the silent engines of our community. Can't do anything without them. They bring their best to the lab or the space every day. They push the boundaries of innovation. They are highly engaged in the discovery process. They train our graduate students. They train our members of the community, and they're highly engaged within the overall community. So I just want to say from the bottom of my heart, as your dean, thank you. All right, Today, of course, we're here to celebrate, and hopefully, we are going to have some fun along the way. And actually, I believe we are going to have some fun because I see all kinds of signs all over this room. And so there will be some celebrating today, which is good. Now, I like to say that why didn't y'all give me a lapel? I feel like I'm chained here. I like to say that, UCSF doesn't have an undergraduate campus, so, we don't have any sports that we can celebrate. But this is like our Pro Bowl or a College Bowl. This is as good as it gets at UCSF. So I want to encourage you to be as vocal in celebrating your favorite person. And even if they're not your favorite person, but you think they're doing awesome signs, I want you to make sure that you are celebrating them all. And I want you to make sure that you are keep in check because you will get a chance to vote, right? There's a people's choice. So make sure you track them so you know who you want to vote for. Now, full disclosure, don't just vote for the person you came here to support. All right, I'm gonna leave it right there. All right, Postdoc Slam benefits not just our presenters because they have learned how to condense some really dense, challenging work into language that just about anyone can understand. It's a very special talent, and a talent that, quite frankly, we need right now, right? Anybody ever talk to a congressperson? Do we have any science policy folks in here? Do we have any science policy? No science policy folks? Speaking to the community is so important for us at this moment. Every year I say the same thing, and every year I say, especially at this moment, and it's like it's true, especially at this moment. It's really important for us to communicate, and they have spent a lot of time, effort, and energy in condensing their work in ways that a normal an educated audience can understand. So we are looking forward to that. 2 points. So in between now and when the judges leave, you are going to have the opportunity to vote. I want you to remember, keep that in mind. And then there's going to be food outside. So now when you get to the food outside, don't just leave, okay? There's stuff that you have to come back for because we're not going to announce the winners until after the food outside. So make sure you stay around for the entire event. All right, I am basically done. I am going to turn it over to your postdoc dean, Dr. Shaquita Allen.
Thank you, McKay. Hello, everyone. Welcome. And I want to take a moment to wish all of our postdocs here at UCSF a happy National Postdoc Appreciation Week, following on the dean's comments. You are the silent engines, and we want to make sure you know you are seen and heard and valued. So thank you all for being here. Today, you're going to hear from 10 finalists across our over 1,000 postdocs are here at UCSF. They're going to be presenting to you. They are our finalists after submitting their video presentations to a committee made up of faculty, staff, and alums that reviewed their videos and decided these were the 10 best finalists. So they're going to be presenting those talks to you. They've all been working really hard, being coached, practicing, and you're here rallying for them, which is great to keep them pumped up. Awesome. But there's money involved, of course. The first prize is worth $4,000. The second prize is worth $2,000, and the third prize, $1,000. And then, as the dean mentioned, there's the People's Choice Award for $750, which you get to vote on. So those of you who are here in the audience, and also those that are watching remotely on the live stream, you'll get to vote as well. So it's going to be an exciting time for everybody. So we also are going to have some trivia questions in between the presentations. And we will be asking you and the audience that's remote to raise your hand, weigh in, answer the question. If you get the question right, Xingyi is here. She's going to be pointing out to those who are first to answer, and there'll be a small prize that will be awarded to the person who gets the trivia question right, okay? We have a broad category of pop culture, potpourri, a lot of different topics that our team has put together, so you're going to enjoy it. It's going to be fun. All right? Now, before we get to the presentations, we want to recognize our esteemed panel of judges who are going to be carefully weighing the presentations to decide who our winners are. So I want to take a minute to just introduce them and ask each one to stand as I call their name. Erin Alday, she's a staff writer for the San Francisco Chronicle, an alum of Postdoc Slam judging panels. So she's an expert, she's a returner, so it's a lot of fun. Uwe Klein, who's a senior vice president, biological sciences at Septerna, a UCSF postdoc alum, executive board member, UCSF Graduate and Postdoc Alum Association. Thank you for being here. Letty McNeil Light, associate vice chancellor for university development and alumni relations at UCSF, president, Strategic Initiatives Group for Advancement Professionals. Thank you, Letty. Juliana Malulu, high school CTE health science instructor within the Health and Behavioral Science pathway in the San Francisco Unified School District and a collaborator with UCSF Science and Health Education Partnership. Thank you, Letty. And then last but not least, Jennifer Nazarino, who's our Associate Dean of Academic Affairs here at UCSF in the Graduate Education and Postdoctoral Affairs Office and a UCSF Sociology PhD alum. Thank you, Jennifer. Okay, so thank you, everyone. We want to make sure that we stay on time. We want to remember our judges have criteria that they're focused on in terms of thinking about how they'll make our selection for winners. Did the presentation help you understand the topic? Was the topic and its significance communicated using language that was appropriate to an educated but not expert audience? And 3, did the presentation make you want to learn more about the particular topic? Each talk will be timed. And we have Doug Carlson here to make sure people stay on time. It will be 3 minutes each. If they go over 3 minutes, they will be disqualified, so they'll be watching the clock to make sure that they stay on time. Doug is an expert because he has a lot of experience. He's come back year after year to help here with Postdoc Slam, but he also was the game clock expert for the NCAA West Regionals in 2022. He is the official timekeeper for Cal football and basketball games. He spots the downs and yards to go for the scoreboard operator at the 49ers games. So Doug, he's a pro. Thank you very much for being here. And finally, everyone, out of respect for our presenters and those who are listening, please silence your phones. No texting, no talking, no flash photography. There's going to be a lot of production happening up here, so we want to make sure our speakers have the best moment possible. Okay? Are you ready, judges? Yes. Are you ready, finalists? Yeah. Oh, come on. You got to have a strong yeah. Are you ready, audience? All right, so let's go. Our finalists do numbers, so we're going to go in the order that they accepted. Our first speaker, Sangeeta Naranjan. Her faculty mentor is Daniel Miner, and the title of her talk is From Hide and Seek to Spotlight, Nanobodies Expose Calcium Channel Secrets.
Imagine your brain being the guitarist and your heart as the drummer. Sounds like a whole concert, right? But calcium channels decide whether it's going to be a rock and roll or your neighbor next door practicing guitar at 2 a.m. So what are these calcium channels? Your each beat, each thought, and each movement rely on the rhythm of charged particles flowing through tiny molecular gates located on the cells like neurons and cardiomyocytes. These gates are called voltage-gated ion channels. And one of them that allows calcium ions to pass through are calcium channels. But why are they so important? Because when they misfire, the consequences can be fatal, causing seizures, arrhythmias, and even sudden cardiac death. Basically, these are master regulators of electrical signaling in the body. And when they misfire, we know that a lot of disease could happen. Surprisingly, we don't fully understand that how these channels are built in the native environment, like the heart and the brain tissues. Therefore, to understand them better, I have pulled them out of the native environment, like the human heart and the human brain. And I'm very excited to tell you how I've done this. To pull them out, I'm using tiny antibody fragments called as nanobodies. And these nanobodies are made in llamas. They bind very specifically to one of the subunit of this calcium channel without interfering its function. With these nanobodies, I have captured calcium channels in action. And I am also able to study its interaction with hidden chaperones like EMC. These are basically involved in the folding and assembly of these channels to make them functional. Exactly like the backstage crew that sets up this whole orchestra to make it function. So, as we know that since nanobodies are helping me to do that, but what are the other things that has helped me to do this study? So with nanobodies based pull down, another technique known as mass spectrometry, I'm able to map different calcium subfamilies across the different regions of the heart and brain tissues. And overall, these insights are helping me to understand that how these calcium channels are built, where they go, and how do they differ in disease. It also opens the path to understand and build new precision drugs that don't just block these channels but modulate their assembly. So since I'm always after heart and the brain tissues, and if your X is still staying in there, I'll be happy to take a slice for science. Honestly, that might be the best use your X has ever been. Thank you.
All right, thank you, Sangeeta. All right, now for our trivia question, our very first trivia question. Adam Summers, then a postdoc studying fish biomechanics at UC Berkeley, advised filmmakers on fish locomotion, how fish scales reflect light, and the mechanics of waves during the production of the 2003 animated Disney film, which was, who's first?
The person with the blue shirt.
Nemo?
Yes, that's correct. Finding Nemo. And I'll read a little bit of information after each answer. The answer is Finding Nemo. So while Finding Nemo takes libraries for narrative and visual appeal, it surprisingly accurately portrays certain aspects of marine life. For instance, clownfish like Marlin and Nemo do live in symbiotic relationships. They live in symbiotic relationships with sea anemones, and they're protected from their stings by a mucus layer. Dr. Summers is now on the faculty of the School of Aquatic and Fisheries Sciences at the University of Washington, where his research focuses on understanding the effects of material properties on the form and function of organisms. Summers has been active in science communications and writes on his lab website. Communicating the research we do in the lab is important. Communicating science at large is more important. Everyone in the lab takes this job seriously, with the proviso that it has to be fun if it's going to get done. Pretty good. Judges, need more time? Okay, they're good. All right, our next speaker, Wei-Chung Lee, faculty member, who is the mentor is Jonathan Ostrom. The title of the talk, Rewriting the Rules of Cellular Quality Control. Wei-Chung.
よしだ僕を連れて進め
Picture the busiest factory you can imagine. The machines humming, products rolling off the line nonstop. That's your cells. And imagine this. There's a teeny tiny screw just rewrite the entire quality control system in the factory. And that tiny screw called BRD 1732. And it changes how we fight disease. Every cell in your body is like a factory that produces tons of products every day. Those products are proteins. To stay healthy, they need to remove the broken or unwanted products. It uses the quality control tag called ubiquitin. Think of it as a red sticker or red flag. Once a protein or product is red flagged, then this one can be taken by the disposal system to out. One rule we know really well is that only the big products, like proteins, can be disposed. Smaller pieces, like screws or small molecules, were ignored completely. But at UCSF, in Jonathan Alström lab, we challenged this rule. From a newly established chemical library, we screened and identified a novel small molecule called BRD-1732. For the first time ever, the small molecule can be seen and tagged by this system. This discovery changes how we understand the inner workings of the cell or the factory. And this is not only about curiosity, it's also a game changer for medicine. Think about this. We can attach the BRD-1732 that can be recognized or seen by the system directly to the harmful protein. Then we made a custom removal tool. On one hand, we have BRD-1732 that the system can recognize. On the other hand, it can grab the harmful protein. The disposal system can recognize and spot the BRD-1732 and take the entire thing out. This allows us to target those disease-causing proteins and then make a new way for medicine. I think the BRD-1732 is not just about small molecule. It's a proof that the cellular quality control system can be rewritten. As a result, I think we can imagine to build a new class of therapies. This entire new class therapies can act like precision tools to take those targeted protein out of the body that we can never touch before. So with that, building on this discovery, we can keep the factory of life running smoothly and then keep us healthier. Thank you.
Excellent. Thank you, Wei Cheng. All right, trivia question. Here we go. Released in 1972, this was the first commercially successful video game. What was the name of it? Yes, what did you say? Pong, that's correct. Got to make these questions harder. Developed by Atari, the very first Pong prototype was placed in Andy Capp's Tavern, a local bar in Sunnyvale, California, in August of 1972. Within days, the machine malfunctioned. It had simply overflowed with quarters from enthusiastic players. The paddles in the original Pong didn't reach the very top of the screen due to a circuit flaw. Instead of fixing it, Atari engineer Alan Alcorn realized it made the game more challenging and prevented expert players from volleying forever, so he kept it as a feature. During the 1975 Christmas season, Atari released a home version of Pong. The home version was also a commercial success and led to numerous clones. Pong is considered to be one of the most culturally significant video games and is part of the permanent collection of the Smithsonian Institution in Washington, D.C. See, you learned something. Awesome. Judges, how are we doing? All right. Very good. Our postdoc finalist number 3 coming up is Logan Leake. Logan's faculty mentor is Trevor Bavona, and the title of his talk is When Worlds Collide, Oncogenic Fusion Proteins in Lung Cancer.
A glass of water, harmless. A laptop, also harmless. But spill that water onto your laptop, and now you've got a big problem. That's the kind of issue we see in some cancers. 2 genes that are normally okay on their own come together in the wrong way. Let me tell you an example from cancer biology, an oncogenic fusion protein called EML4-ALK. On their own, EML4 and ALK are just normal parts of the cell. EML4 helps with the structure. ALK helps certain cells to develop in the right way, especially in the brain. However, sometimes there is a genetic accident that happens, and the cell fuses these two together. Now, EML4, which we can think of as our water, and ALK, which we can think of as our laptop, become a single fusion protein, EML4-ALK. And just like the example with the laptop, this can lead to all sorts of trouble. Water can cause a laptop to short circuit. Similarly, EML4-ALK can rewire signaling in the cell. Now, EML4-ALK fusion can rewire it, and this can lead to issues within the cell. Now, sorry, one second. EML4-ALK is an oncogenic fusion protein, and it can cause rewired signaling in the cell. This can lead to a type of cancer called lung adenocarcinoma, not because the individual components were dangerous, but because the combination was. And you might wonder how this is happening. Our lab discovered that these EML4-ALK proteins can form these clumps in the cell. These clumps can turn on the growth signals even when it's not supposed to grow in the first place. So, how do we solve this issue? Let's take a look at the example of the laptop. Some smart people realized that you could remove water from the laptop by soaking it in rice, which can get into tight spaces and readily absorb water. In this case, better understanding the problem was the key to finding the solution. My research focuses on better understanding these EML4-ALK clumps so that I can identify better ways to intervene, treat patients, and reverse the disease. In order to do this, I conducted a proteomics analysis to identify proteins that are present at these clumps. I discovered that certain proteins that don't normally interact with EML4 or ALK are present at the clumps. And I'm currently working on a strategy to drug these proteins in order to reverse the disease. I'm excited to continue testing these in preclinical models. Now today, I presented to you two very different anecdotes, but in both cases, the story is the same. Two things that are harmless on their own can come together to cause a really big problem. And in both cases, better understanding the problem is the key to finding a solution. Thank you.
Great. Thank you, Logan. All right, our next trivia question. What four-time Pulitzer Prize-winning poet, mostly associated with New England, was born in San Francisco in 1874? Gotta make it tougher for you. Take a guess. Yeah! Great guess. All right, really good. Robert Frost, the famed American poet who was best known for writing verses about rural New England life, spent his formative years living in San Francisco, in the city, with his family. Upon his father's death in 1885, his mother moved the family to Massachusetts. Reportedly, Robert was a street kid. He hated school and manipulated his mother into letting him stay home much of the time. She taught him herself, but he resisted her. He was lazy and homework, he didn't like to read. He was lazy about his homework and he didn't like to read at all. Imagine that. At the other end of his life, Frost was the first poet ever to read at a U.S. presidential inauguration for John F. Kennedy in 1961. In the decades since, inaugural poets have included Maya Angelou, Miller Williams, and Amanda Gorman, among others. Today, Robert Frost Plaza sits on Market Street at the terminus of the California Street cable car line, marked with a historical plaque. How about that? Great. All right, judges, thank you. Our next presenter is Atreya Day. Atreya's faculty mentors are Andre Shali and Ignacia Echeverria Riesco. The title of his talk is A Theory of 100,000 Grandmas.
When I was a child, I remember my grandmother used to love knitting. And something about the pretty colors and the soft texture of the yarn was so appealing that whenever I used to get anxious, I used to sneak out a ball of yarn and played with it. Inevitably, the yarn would get all tangled up, and my poor grandmother had to spend hours untangling the mess I had made. Well, tangled string can be a nuisance in knitting, but did you know that tangled string can also give you cancer? Okay, so the human DNA is a lot like a string. In fact, the DNA in the human genome is a 6 feet long string, and all that is tightly packed inside our tiny cells. And every time a cell divides, there's someone like my grandma sitting in the cells untangling the DNA. This tiny grandma untangling the DNA is a protein called condensin. And understanding how condensin does this is crucial for preventing diseases like cancer or Down syndrome. So when I started this research, we had no idea how condensin works. Over the next few years, some experimentalists came up with some results on how condensin might be working. So the first experiment told us that condensin looks like a ring, and it can burn energy to pass DNA through that ring. The second experiment told us there are 100,000 condensins present in a single cell. The third experiment gave us a bird's eye view of what that 6 feet long genome looks like when condensin has neatly packaged it right before cell division. So I'm a theoretician. Using all these clues, I made a theory called the theory of 100,000 grandmas. Using my own grandmother as a reference, I assumed that condensin is like a grandma knitting loops out of DNA. When they get tired, they simply stop working and leave. When they come back, now they're grandmas. They don't remember where they were working, so just start knitting at a random spot on the DNA. When two grandmothers try to knit at the same spot on the DNA, they both get into an argument, they both stop knitting for a while, and then they continue knitting as is. So using this theory of 100,000 grandmas, not only were we able to recapture the behavior of single condensin molecules, we were also able to get that whole bird's eye view picture of the 6 feet long genome. We believe this theory is the first steps towards understanding condensin-related diseases. Now, it's been a while since my grandmother has passed. But even then, when I feel very anxious, like when I'm up on a podium talking to an audience, it feels nice to know that there are 100,000 grandmas in my cells protecting me from cancer. Thank you.
Thank you, Atreya. All right, now for our next trivia question. Which street in San Francisco is actually curvier than Lombard Street's famous crooked section, featuring 7 sharp turns within a single block? What's the name of the street? What is it? I can't hear you. Vermont? Yes, that's correct. Yeah, you have to shout because we don't have microphones right now. Let me tell you a little bit about Vermont Street. Tourists flock to the famed Lombard Street, long thought to be the crookedest street in the world. But the San Francisco Department of Public Works has confirmed that this section of Vermont Street has a higher rate of sinuosity than Lombard. Tucked away in Potrero Hill, Vermont Street is a relatively well-kept secret, although it was the settling for a chase scene in the 1973 Clint Eastwood film, Magnum Force. It's also the site of a raucous and colorful race seen each year on Easter Sunday, when dozens of daredevils participate in the Bring Your Own Big Wheel event, in which they careen down the curves to the cheers of hundreds of spectators. Any takers? All right. Okay, great. Our next presenter, we're halfway there. Jack Moen, whose faculty mentor is Nevin Krogan. The title of his talk is From Beast to Bedside, Animal Biology That Heals. Jack.
We've all seen the headlines about miracle cures, life-saving treatments just around the corner. But for many, the promise never reaches the pharmacy. In fact, it now costs up to $3 billion in 15 years to bring a drug to market. Our current approach to drug discovery is slow, inefficient, and broken. But what if solutions to some of our toughest medical challenges already exist? They're just not in humans. Animals have evolved over millions of years to fight infections, heal wounds, and even edit their own genes. We believe that by studying these biological superpowers, we can shortcut the drug discovery process and bring therapies to patients faster, cheaper, and better. Take, for instance, the alligator. These animals live in swampy, bacteria-filled waters, yet they can shrug off the most gruesome wounds. While studying this effect, we applied alligator serum to cells, and we noticed something incredible. It caused the cells to become more stem cell-like, making them flexible and regenerative. We're now trying to isolate the molecules behind this effect with the hope of developing therapies for slow healing wounds, burns, and surgical healing. Next, consider the squid. These animals do something remarkable. They're able to edit their RNA without modifying their DNA. This gives them the ability to fine-tune protein production in real time. So why does this matter? In cold waters, chemical reactions slow down. In response to this, squids edit their proteins to speed up their function. This allows them to keep their reflexes fast and their systems sharp, even in freezing temperatures. We're now studying this naturally occurring RNA editing system with the hopes of developing therapies for treating genetic diseases in humans without having to modify DNA. Lastly, consider the turtle. Not only do these animals live long, they also fight infections with a novel immune system. Whereas human antibodies are rigid structures that bind to a single target, turtle antibodies are modular and flexible, able to bind to divergent targets that may be far apart and moving. By studying turtle antibodies, we hope to create systems that are more adaptable and flexible, that respond with the same sophistication that nature's already perfected. So while our current approach to drug discovery struggles, the solutions to our toughest medical challenges might already be right in front of us, and the animals that have figured it out. It's time we stop reinventing the wheel and start borrowing from nature's playbook, because healing might not always begin in a lab. It might begin in a swamp, a reef, or even a shell.
All right, thank you, Jack. Okay, we're ready for the next trivia question. What is a group of flamingos called? No.
Oh, wait, sorry, flamboyance.
Yeah, that's it. Okay, we gave you 2 guesses, but that's okay. All right, let's learn a little bit about a flamboyance. All right, there are other terms for a group of flamingos, mostly commonly a stand, but we can say we're looking for this specific one, so it was flamboyance. The collective noun to describe a gathering of flamingos is flamboyance. It's an appropriate term for these colorfully feathered creatures. They flock together by the thousands on salt flats, lagoons, lakes, and swamps around the world, where they can filter feed for shrimp, algae, and insects. Both flamingo and flamboyance come from words referring to fire. Flamingo comes from the Spanish and Portuguese, literally flame-colored. Flamboyance comes from French, meaning to flame or flare. In the U.S., flamingos of the pink plastic variety can be found as kitschy lawn ornaments. They were first designed by Don Featherstone in 1957 and quickly spread in popularity. Featherstone won a 1996 Ig Nobel Prize in art in honor of his ornamentally evolutionary invention, now gone away. Flamboyance. Okay, great. Ready for our next speaker. Our speaker number 6, finalist, Da Zhang. Da's faculty mentor is John Clean, and the title of her talk is AI-Powered Epilepsy Detection, Bridging Gaps in Care.
When I was a kid, I saw one of my friends suddenly collapse in front of me. It was terrifying. And after that day, I learned a new word that I never heard before, epilepsy. I didn't know what it meant, but I do know the seizures in patients with epilepsy put their lives at risk and make them being discriminated. That life experience stayed with me for the rest of my life and set me on a path to understand the mechanism behind the disease and how we can better treat it. However, after I further dig in, I find out an even shocking reality. In the US alone, there are 3.4 million people battling with the disease, and we only have 2,400 specialists to treat them. That's only one doctor for every 1,400 patients. And this huge gap leaves so many patients' lives being interrupted, and their treatment being delayed, and their family member were left out searching for answers. And this is where artificial intelligence comes in. I developed a system based on vision transformer models, a type of AI that sees the brain waves the way you and I recognize a dog in an image. So for example, given the image of the dog, what is the first thing that you pay attention to decide it is a dog? The eyes, the snout, the ears, right? So all those features interact with each other spatially and let us make the decision it is a dog, even without further thinking. And similarly, our AI model learns the hidden patterns in brain waves that indicate seizures, the hidden pattern that even experienced doctor might miss in real time. And even in testing phase, our AI model achieved expert accuracy across patients, even on cases that it had never seen before. And why this matters? That means faster and earlier diagnosis, more patients getting care, and fewer people facing the crisis my friend went through. By the way, we are not replacing doctors. We are just giving them a powerful tool to reach out patients who need help. Lives will be saved, and no child has to watch their friend suffer again. Thank you.
Great, thank you, Dazhang. All right, we're ready for our next trivia question. This day in history, NASA unveiled its first space shuttle on September 17, 1976. What was the name of it? Guesses? Any guesses? Need a hint? Okay.
Discovery?
No? No? You're on the right path. No?
Columbia?
No? No? Okay, we'll give you a hint. It shares a name with a famous fictional spaceship. Yes, Enterprise. On September 17th, yeah, that's good, 1976, NASA publicly unveiled its first space shuttle, the Enterprise, during a ceremony in Palmdale, California. Development of the aircraft-like spacecraft cost almost $10 billion and took nearly a decade. And no, you're not seeing things. This photo shared by NASA in 1976 shows some familiar faces from another Enterprise. Cast members from Star Trek were on hand for the unveiling of the space shuttle. You might recognize Dr. McCoy, Lieutenant Sulu, and Uhura, and Mr. Spock, among others, for you Trekkies. In 1977, the Enterprise became the first space shuttle to fly freely when it was lifted to a height of 25,000 feet by a Boeing 747 airplane and then released, gliding back to Edwards Air Force Base on its own accord. NASA's final space shuttle mission came to an end in July 2011. All right. Our seventh speaker coming up, Jareen Tasneem. Her faculty mentor is Xinhua Piao. And the title of her talk is Breaking the Silence, How Astrocytic GPR56 Can Protect the Alzheimer's Brain.
No, I didn't forget my speech, but that's exactly how it feels when you lose your memory. That uncomfortable silence, confusion, emptiness. That's the reality for millions of people living with Alzheimer's disease. And what is worse, we still don't know how to stop it because there is no cure for Alzheimer's disease till death. Our brain does try to protect itself. It has a built-in support system we call glial cells. Two of the most important glial cells are microglia, the cleanup crew that removes all the toxic buildup in our brain, and astrocyte, the caretaker that helps everything run smoothly. They are like teammates, working together behind the scene to keep our brain functioning. But in Alzheimer's disease, this teamwork falls apart. In our research, we actually found something remarkable. Our receptor called GPR56 sits on astrocyte, and it works as a communication switch. When we turned off that switch by deleting that gene in an Alzheimer's disease mouse brain using a gene editing tool called CRISPR, everything got worse. We saw more toxic buildup or more amyloid plaque, less cleanup, and more neuronal damage. It was as if the astrocyte couldn't call for help anymore and microglia didn't know where to go. These results clearly suggest that GPR56 is playing an important role in protecting Alzheimer's disease brain. So now we want to know more. We want to know exactly how this communication is happening between GPR56 on astrocyte and microglia. For that, currently we are growing human cells in a lab dish, and by using proper gene deletion or gene mutation, we are creating an Alzheimer's disease microenvironment in that lab dish. We are also using advanced tools like proteomics, which is basically a molecular snapshot to see how the proteins are interacting or how they are changing to understand the cellular conversation. So, what's the point of it? If we can understand this cellular conversation or decode this cellular talk, we can identify a new and proper drug target to cure Alzheimer's disease. So, we are not just studying cells in a lab dish or in a mouse model. We are working to end that terrible silence. We are fighting to bring back those voices, those laughter, those stories that made us who we are. Thank you.
Thanks, Jareen. Thank you for that. Okay, next trivia question. Long before Black Rock Desert, the Burning Man Festival was born at what San Francisco site? Do another.
1, 2, 3. The person with yellow.
Yeah, Baker Beach. Yeah, raise your hand first. All right, Burning Man was born in San Francisco as a bonfire on the summer solstice. On June 22nd, 1986, while the Presidio was still an active Army post, a new tradition started at the northern end of Baker Beach, Burning Man. Relatively isolated and full of driftwood, the north end of the beach was an ideal spot for a group of 20 friends to gather for a bonfire where Burning Man founder Larry Harvey set aflame the 8-foot-tall wooden structure he called the Man. Burning Man relocated to the Black Rock Desert in Nevada in 1990. Word of the festival spread by word of mouth in the early years, and we all know where that eventually led. These days, the gathering draws over 80,000 people, and the Man typically stands over 70 feet high. Judges? Okay. We're ready for our next speaker, Leah Lordo. Her faculty mentor is Hitan Madani, and the title of her talk is Cracking the Genetic Code of a Deadly Fungus. Leah?
Fungal infections are incredibly common. They affect 1 in 8 people around the world. That's 1 billion people that have a fungal infection right now. Of course, some of them are mild, like nail infections, but others are life-threatening, like blood infections, and they kill 3.8 million people every year. Treatments are limited. Current antifungals don't always work, they can be toxic, and fungi are becoming more and more resistant. And right now, there are no vaccines available for any fungal infection. That's why studying fungi is essential. In 2022, the World Health Organization released its first report on the most dangerous fungi, and at the top of the list is a brain-invading fungus called Cryptococcus neoformans. That's the one we study in the Madani lab here at UCSF. Cryptococcus neoformans is found in soil and bird droppings, and most of us breathe it in at some point and don't even notice. However, in people with weakened immune systems, like those living with HIV and AIDS or those who have organ transplants, it can cause dangerous lung and brain infections. What makes Cryptococcus so dangerous is that it can grow at human body temperature and beyond, too hot for fungi normally. And this ability is called thermotolerance. But we still don't know all the genes that are involved in thermotolerance. So in the lab, I use a powerful gene editing tool called CRISPR. It works like molecular scissors that can cut genes one at a time and switch them off. Using this method, we can turn off more than 5,500 genes in Cryptococcus, creating a giant library of mutants. Then we can take this library of mutants, put it through a heat challenge, and see which mutants survive and which ones don't, telling us which genes are involved in thermotolerance. But just like viruses like COVID have different variants or strains, Cryptococcus also has different strains, and it's important to study them all. So the goal of my project is to make these giant library of mutants in the different strains and then put them all through a different heat challenge. And by comparing cross strains, this will tell us which genes are always important for thermotolerance across many strains, and which ones only matter in specific strains. And then, once we identify these genes, this will inform us more about fungi, and it will give us clues to develop more precise therapies. The better we understand fungi, the better we can fight them. Thank you.
All right, thank you, Leah. All right, next trivia question. What famous metal singer, best known as the frontman, probably a lot of you know this, of Black Sabbath, had his genome sequenced, revealing fascinating insights into his unique genetic makeup? Ozzy Osbourne. Yes, Ozzy Osbourne. Also known as the Prince of Darkness. Ozzy Osbourne, who died this summer at age 76, was a musician, first with Black Sabbath and later with his own band, and eventually a reality TV star with his family in MTV's The Osbournes. He also had his genome studied. In 2010, biotechnologists approached Osbourne about sequencing his DNA to investigate the genetic factors contributing to his survival despite decades of substance abuse. Osbourne was skeptical. The only gene I know anything about, he said, was the one in KISS. But he agreed when the scientists convinced him the results could help explain how he survived 40 years of intense drug and alcohol abuse and all the ill-advised antics that go along with it. The analysts revealed novel genetic variants and segments of Neanderthal DNA, similar to what is found in other people of European descent. Specifically, they found a mutation near the ADH4 gene, which is involved in alcohol metabolism. This mutation is potentially linked to how his body processed alcohol, led to his survival. Don't try this at home. Okay. Next presenter. We have two more. Our next presenter is Rabab Rangwala, and her faculty mentor is Srikantan Nagaranjan. The title of her talk, Where There's a Swallow, There is a Synchrony.
Every breath you take, every move you all take a pause and let's think about our favorite food. Maybe you like ramen from Japantown, or if you are like me, we like a delicious slice of cake from Tartine. Food is a source of comfort, and it's a trigger of joy. And every time we relish it, there's a quiet synchrony happening right within us. If you have some water, please go ahead and take a sip. If not, just swallow, go ahead. Did you feel it? A tiny automatic pause in your breath. You're breathing and you're swallowing in the perfect symphony, very much taking a cue from the brilliant mastermind conductor, your brain. Now you know what I do all day. I study swallowing. I'll share an honest confession. It's been 10 years in this field, and people still think I'm making a dirty joke when professions are discussed. In healthy adults, the rules of this orchestra are very clear. We exhale, we pause, we swallow, and we exhale again. But in patients with Parkinson's disease, this rhythm falters. Breathing and swallowing fall out of sync, and food and liquids tend to go the wrong way, marching towards the lungs. This is what we call aspiration. Food can go from a source of joy to fear, and even a dangerous form of pneumonia, a leading cause of death in patients with Parkinson's. But here's the promising news. In my world of research, we are the music teachers, helping patients find their rhythm again. We use tiny wearable sensors that go on the base of their throat, and we record every breath and every swallow. We then give our patients simple swallowing cues in form of tiny haptic vibrations, helping them relearn the 1, 2, 3s of swallowing. Because this isn't just about swallowing. It's about being able to take a sip of water. It's about being able to enjoy ramen with your loved ones. It's about restoring dignity, joy, and connection. One breath, one swallow, and one life change at a time. Thank you.
Thank you, Ava. All right, our next trivia question, here we go. Vice Admiral Robert Fitzroy coined this term for his pioneering data-driven weather predictions, the first ever published in a newspaper in 1861. What was it? I can't hear you. No. Specific term. Yes, forecasts. You may know that Robert Fitzroy was the captain of the HMS Beagle on Darwin's famous voyage to Tierra del Fuego, but in his own lifetime, Fitzroy was best known for making daily weather predictions, which were published in the Times of London. Fitzroy was troubled by the massive loss of life at sea along the coasts of Victorian Britain and believed that forewarning ships about bad weather could save many lives. Dedicating himself to this cause, he directed the installation of barometers at every British port where crews could consult them before setting out to sea. He also used the newly established telegraph network to gather at his office in London real-time weather data from observers on the coast. If he thought a storm was imminent, Fitzroy would telegraph the threatened port, where a warning drum was then raised in the harbor to save more lives. Okay, and now our final presenter, Ritwik Vatsayan, whose faculty mentor is Prasad Shirvalkar. The title of his talk is Chronic Pain, Is It All in the Brain? Ritwik.
We've all heard those unbelievable stories. The athlete who pushes past a torn muscle to win an Olympic medal, or the mountain climber trapped under a boulder who cuts off her own arm to survive. We are told these are examples of mind over matter, that if your will is strong enough, you can overcome anything. Almost casually, people say, pain is all in your brain. But what does that really mean? Is it just an inspirational quote to put on a coffee mug? Or is it perhaps something more fundamental, a truth that neuroscience is only now beginning to understand? Because for millions of people around the world, pain is not just a metaphor, it is a daily reality. But what if pain isn't something that simply happens to us, but is something that is actively created by us, by networks in our brain that amplify pain sensations long after the body has healed? My research focuses on how chronic pain, which is pain that lasts longer than 3 months, disrupts the communication networks in the brain, and how we can fix these networks. Like pinging a computer in a network, I use single electrical pulses to trace how signals travel through the brain and pinpoint where things go wrong in chronic pain. Normally, pain signals travel from our body to the brain through pathways that both sense and respond to injury. But in chronic pain, we have found that these pathways remain active long after the injury has healed. Like muscles that get stronger with repeated workouts in the gym, these circuits become overactive, amplifying sensations with no physical cause. At the same time, the brain's higher regions, such as the prefrontal cortex, which regulate pain and emotions, get overworked. They're called on too often and begin to wear down, losing their ability to effectively regulate pain. This creates a vicious cycle in chronic pain. The sensing part gets stronger and the controlling part gets weaker. It's like a car with a stuck accelerator and failing brakes. But here's the good news. Because the brain is plastic, meaning it can change and adapt, we can attempt to reset these networks. By identifying dysfunctional networks in the brain, I use the same electrical pulses to zap the brain back into balance, providing therapeutic relief without having to rely on addictive opioid medications. Think of it this way. While painkillers might tap the brakes, they do not fix the stuck accelerator. My research aims to tackle both, restoring balance to the brain's networks so that our patients don't just mask their pain, they truly regain control of their lives. Thank you.
Warm round of applause for their amazing work. Yeah. Outstanding. Great job in the lab, great job today. Congratulations to all of you as finalists. You did an amazing job. Another round of applause for them. All right, as they go back to their seats, we're going to prepare for the judges to do their final calculations. They're going to be whisked away. You can go back and be seated. And join everyone in the atrium while the judges are calculating their marks.
We are back. All right, how was the food? How was the food? Not bad, not bad. All right. Thank your dean. That one, not this one, that one. Thank your dean. All right. I want to start. I always get this wrong, so I decided this year I'm just going to read my notes instead of ad-libbing because I just always mess it up. So in an effort not to mess it up this time, I want to thank a few people before we get to the grand finale. So I want to thank our 5 judges for their hard work. Give them a round of applause for me, please. Thank you. Now, did you enjoy the presentations? Uh-huh. Well, all that hard work comes with a lot of practice, and We have a young man in our shop that helps the contestants to refine their talks. So give it up for David Blanchard. He is not here. He's watching online, but he's in our Career and Professional Development office, and he does a fantastic job. Now, the people who get the most blame when things go wrong and get no credit when it goes right, and for this group, it always goes right, is our ETS guys. The guys who are doing this, who are videoing, doing all of that stuff. Give them a round of applause for me, please. Now, I think you know that an event like this takes a lot of planning and a lot of work. I don't do any of it. But there are people in my office who really work hard at putting it together. So looks like they're all outside because I can't see any of them in here. Eric, there he is. Come here, Eric. Show your face, Eric. That young man. Let me tell you something about that young man. He's a special dude. He is the one that figures out all the trivia over the last 8 years, and he makes it fresh. Who knew there was so much history in San Francisco? My God. Like, who would have known all of that? But Eric does a lot of the calm side of the planning, and if you like the food and the event itself, that's our event planner. That's probably not a good name for her, but it's what she does. Annalisa, I don't know where she is. She is somewhere around, but we can't do this without her. So give Annalisa a round of applause. But this doesn't happen just with them. It's really all hands on deck from the GEPA folks. So thank you all for the GEPA and the OESA folks for your help. Round of applause again. And then my last one is, how much was the first place award again? Yeah, we know that, huh? Don't we? It's $4,000. And for this year, for the first time for Postdoc Slam, we have a very, what do I say to Sam? We are a very generous donor that gave us the money for the $4,000 prize. So please give it up for Sam and his beautiful wife. All right, now are you ready? So we have 4 awards. I have them in my hands. We are going to ask the judges, some of the judges, to make the awards. The first award goes to your choice, the people's choice, and we're going to ask Juliana to come up and make that presentation. This is $750. All right. And the winner is, drum roll please. Yes.
Rabab Rangwala.
You stand there and you can hand it to her. Rabab, hand that to her. That's not real money. But by magic, real money will appear for you. Okay. All right. Thank you both. Oh, hold on. He's doing some photographs there. All right. And now we're calling up our judge, Letty, to come and present the third place award for a full grand, 1G. All right. Yes, you can. And the winner is...
Delighted to say that it is Rabab again.
That's not a bad payday, Rabab. Research pays. Okay, it's only $1,700. Let's settle down, okay? All right. Our second prize, we're going to ask Uwe to come and make this presentation. There you go. You know the drill. And the winner is... Keep going. We're going. He needed his glasses. There we go. There we go. We're coming. We're coming.
And the winner is Darren with GPR 56.
Oops. Say that again.
Darren, GPR 56.
Woohoo!
Congratulations.
Picture. Yay. Excellent. Oh, just so you know, that was 2 G's. 2 G's. Not bad, 2 G's. All right. And now for the grand prize of $4,000. Let's invite Sam up to do the last presentation. All right. Yes. For now. Okay, yeah, gotta use the mic to make the announcement. Oh my God. Sam, use the mic, Sam, use the mic, yeah, for the live stream.
The top prize winner is Atreya Day.
And you get a big check. Don't take it to the bank. All right, take a picture with Sam now. That check is going nowhere. Like, this is my check. Thank you very much. All right. Congratulations to all the winners, but seriously, congratulations to all the finalists. You did an amazing job. Thank you. We will continue to celebrate National Postdoc Appreciation Week. Look out for the events. I hope that you will, if you're not a postdoc, that you will take a moment just to say thank you to your postdoc when you go back to lab sometime this week, especially this week. You should say thanks all the time, but just this week, as a special, extra special thank you. To the postdocs, I want you to know that we see you. We hear you. We know that it is tough times, but we also know that your curiosity, your dedication, your expertise are really the driving forces behind so much of the innovation and discovery that happens here at UCSF. So, yeah, that's not for me, that's for you. So from the bottom of our hearts, thank you so much for all that you do. Have a good evening, everyone.